unit, so last bit of the semester, and it's going to put into application what we've learned so far. So it's, as I mentioned, basically every unit this course is a building course, and so we're just putting the pieces together. And then so we're kind of culminating the semester with probably what may be more interesting to you as a student, in that we look at inheritance patterns and how traits are passed from one generation to another. And then we'll actually round out the semester with a very kind of hot topic with evolution. So we'll kind of give you a biological view of evolution so you can make your own decisions about whether you believe in a scientific theory or not. So in order to get there, though, we need to first look at inheritance patterns, which basically we'll talk today with a review of our Unit 3 material. So it'll be a little bit of a review, and then we'll put it into practice, better practice on Tuesday. After our presentation, we'll go over the exams. I've just graded them, so I haven't even time to look at, analyze the data. But just like past us, the distribution was wide. I mean, it was just crazy. Again, how broad the distribution was. So some were perfect and some were not perfect, and far from average. So we got a huge distribution. So we'll just average is okay. So we'll go over that at the end of the class. And then at the end of class, I will give you your 11th handout that you guys will return to me on Tuesday. It's going to make you kind of think forward, because we're going to introduce something called a Monohybrid Cross, which you've probably heard of a Punnett square before. But this one's going to push you and make you do a dihybrid cross, where you're not doing one trait, but how many traits? Two traits. And so I want you to work on that over the weekend. So the discussion on Tuesday will go a little bit smoother. And then I hope I will have for you the rest of the Chapter 10 handout to get ready for Tuesday. So this last unit will cover Chapter 10, 11, and then we'll combine 12 and 13, which is dealing with speciation and evolution. So we'll put those, kind of consolidate those into like a week with experience. Just to remind you on your handouts, there's obviously the objectives, which is just basically a study guide. So there is a study guide that's been available. So again, my weekly courage. Now that we're coming off from the test, use the study guide, use your handout, prepare for the upcoming exam, not just the night before the test, because that is not an efficient way for the average person to do well. Okay. All right, so we're going to look at inheritance. I found this image a couple of years ago, and I think it's pretty funny, but it kind of gives a good idea about genetics, basically, and traits that we can kind of see. Right, so what is this image? Mom and Dad. It's two different types of sports. One is functional, one not so, a ladle. But, do what? Does anybody like the, in your family, the kid coloring on the wall? The champion of the family. If it's not your sibling, right? So here just have presented to you the objectives. They're listed for you in your handout. Today we're going to just reiterate the role of chromosomes and inheritance. We'll be utilizing these terms called dominant and recessive. And we'll reiterate again what a gene is and apply the term of what an allele is. And of course review what the term chromosome is. We'll also introduce some terms that may be new to you, including genotype and phenotype. And we'll also talk about traits that are homozygous or heterozygous. And those might sound a little familiar, but we'll hopefully clarify that for you. This all ties back into meiosis, because we're looking at inheritance patterns, which is production of offspring, which clearly is going to be the fusion of gametes, whether it be a human, or like we'll study today, plants, or a dog in your yard, or whatever the case may be. And then we may not get to the Punnett square today, but certainly we'll get there on Tuesday about what exactly a Punnett square is and how to utilize this simple tool. And then next week, we'll do the dihybrid cross, and we may do some genetic mapping, or we might call it pedigree, and just kind of review what those represent. And then if we have well, we'll certainly talk about dominant alleles, recessive alleles, incomplete and codominant traits. If we have time, which we may not, which I'm okay with that, talk about pleiotropy, X-linked traits, and those kinds of additional factors that are interesting, but the applied part. We can just understand the basics, then we can hopefully apply it a little bit better. So let's go through and review some of our terms. Okay, so I've got them listed out here, and I'm going to go out of order, just trying to illustrate for you something. And there's a website, and if you want to write this website down, I have it in a large font, top right-hand corner, www.ncbi.gov. That stands for National Center for Biotech Institute. And this website is chock full of information. Active research scientists utilize this program on an hourly basis. Okay, so it's just pinged all the time. There's so much data in there. But there's additional just basic educational tools that you can go and check out. So this image is from the NCBI website. I went here because it illustrates the different chromosomes that would be found in a human. So we're looking at specifically the human, but it has other species. Well characterized are pigs and mice and rats. Think about some other animals that are research-based animals, and their chromosomes have also been mapped. So first thing is, this image illustrates for us how many chromosomes are there in a, how many chromosomal pairs are there in a human? 23 pair. Okay, you'll notice that it doesn't count the 23 because the 23rd is either an X or a Y, and then a pair, either an X or a Y. So collectively, if I'm looking at just, I can see just one chromosome per illustration here. So if this had come from a cell, and we just had one of these, X or Y, what type of cell would that be found in? A germ cell or a sex cell or a gamete or a gene. How did you know that? Those are haploid cells. Okay. If this were visually representing a diploid cell, how many? Two of each. Two chromosome number 1, two chromosome number 2. Would they be genetically identical to one another? Correct. Yes. Of the matching chromosomes 1 and 1, they would not be genetically identical because one half of your chromosomes come from your mom, and the other chromosome comes from the mother or father. Excuse me. Okay. However, where I think you were going is within the cell. Yes. What they would produce during replication would have the same combination. But within the individual, if I'm comparing chromosome 1 to the mother to chromosome 2 to the father, there's going to be some variation. Hopefully. Okay. There's going to be a lot of similarity because both of your parents are what? Human. Okay, so the high proportion of those nucleotide sequences will be identical. But there will be some unique traits that we can see that are different, like most common, like skin color, hair color, eye color, right? Those tangible things, height, etc.
So would they be
That would vary. Okay, so and we'll go there. It would depend on the trait, if the trait for a particular gene. Okay, so let's go back and let's review the terms so we can apply that. So collectively, if this were doubled up, that all of these chromosomes would represent an individual what? Genome. So let's redefine genome in our handout. How can you define a genome? In a nucleus, and more specifically of an organism. Okay, so you're looking at the entire set of genetic material in a particular organism, is its genome. The genome is the entire collection of DNA in an organism. And the genome? Entire set of genetic material or DNA in an organism. Okay, we've been focusing only on the nuclear DNA, but we also know what else has DNA in our human cell. What does that stand for? Mitochondria also has DNA. It's not near as extensive as the nuclear DNA. All right, so that was actually a question on your test. Question number 32, the first fill in the blank. All the genetic material for organisms is known as its genome. We covered that in chapter 7, I think. Okay. So we can look at the genome collectively, or we can look at an individual what? Chromosome. So how can I define a chromosome?
A single strand of DNA with protein?
Yes. Okay, so it's a single. I'm gonna modify the. It is a single unit of DNA associated with protein. The word I'm going to edit is strand because within a single unit of DNA, how many strands is it? Two. Okay. So we're just going to edit that one word. Okay. Strand. Be technical, but it's kind of important.
Okay.
But you're right. So a chromosome is just a single unit of DNA with its associated packaging proteins. Okay. So we understand that chromosomes just a continuous set of double-stranded, anti-parallel, complementary DNA that can be isolated and condensed. Okay? What is within the chromosome? How could we further categorize a chromosome? What is making up a chromosome? Lots of different genes. The chromatid would be a single unreplicated one of these. So within a single unreplicated chromosome or chromatid, what are those nucleotides actually coding for? Individual genes. Okay, that code for proteins, yes. Okay. So if we look at a single chromosome, within a section of a chromosome, a sequence of nucleotides code for individual genes, G-E-N-E-S. It's not your blue genes, right? Your genes. So let's close this out here. Does a single chromosome have a single gene coded for it? Or do we have only 23 genes? No. There may be hundreds or I don't know about, probably thousands of genes coded on a given chromosome. So genes are just sequences of DNA strand that code for proteins. Genes are just strands of DNA that code for proteins. And maybe, right, thousands is a better accurate guesstimate of the total number of genes per chromosome that are coded for. I'd like for you to address that not all genes are expressed. So we started out with Unit 3 with. Not all genes are expressed all the time, right? We don't need to express the protein for bone formation in our teeth. Okay. Or, right, we could just go on all the crazy, weird, mutated kind of scenarios. Okay? So even though our cells have the ability, because the cells in my cheek have the exact same genome as the cells in my teeth, and that make the teeth, right? They're just expressing different genes. Does that make sense? So we have different genes. What are some examples of genes that might express so your keratin in your hair. What else? The color, the pigmentation or eye color. Good. Height. The proteins involved with growth, so hormones and muscle proteins, etc. Actin, myosin. Okay, good. What else? Name some proteins that you labeled on test number 3. For DNA replication. Methionine would be an amino acid, and that is not, it would be coded. A lot of our amino acids come from our diet. However, what would carry? Bind to the amino acids. The ribosomes. So ribosomes have to get made somewhere. So there's a code to make them. Okay, so part proteins. There's some other proteins that we use. Serine, yes, that's an amino acid. It's used to build our proteins. What about DNA polymerase enzyme, which is a protein? So if I need DNA polymerase, I first have to code for it and then transcribe and translate it. So a lot of times we like to think about the tangible proteins like hair color and etc. But basically everything that's an enzyme or protein that we've discussed before is coded for in a specific gene found on a specific chromosome. Thanks to technology, these chromosomes and gene locations have been mapped. And so now we can actually see if a person codes for a particular gene, or if there's a mutation in a gene, if it works or doesn't work, or whatever the case may be. I pulled up some examples of different types of alleles. This is going to be new to us. I think the term was introduced in the last unit, but I didn't use it. So what is an allele?
Different alternative forms of genes.
Are variations, or different alternative forms of genes, or variation of a gene. Very good. What that means is, everybody here, I can see, has proteins to code for hair. Okay? But are all of our proteins expressed the same? No. And so that means is basically some hair is darker, some hair is lighter, some hair is longer, curly, short, whatever. So even though we have code for protein, the keratin, there's variations in how it's built together. Okay, so for example, that I have here, an example is eye color pigmentation. Okay, does everybody have the same color of eye pigmentation? No, but we code for eye color pigmentation. And so the gene codes for eye color. The allele would be blue coding or non-blue coding, as they say. Does that help?
So the alleles are different.
Yes. Yes.
So does that mean like sequences are different?
Yes. Let me read. You're on the right track. I want to clarify. The alleles may be different. Okay. So if you look at all people with brown colored eyes, the code is the same. But if you look at the people with all blue colored eyes, the code is the same. Okay. So they may be different if the proteins just technically. But things that are more conserved to deal with metabolism, like glycolysis, and hopefully all those enzymes are built exactly the same. Okay, so pyruvic acid, all those enzymes involved with that should be the same. Where we get into differences is if they're expressed or not, but the code should be the same. But, yes?
Can an allele play a part in mutation?
Alleles can be mutated, and that's how we get different expressions or combinations. Did you have something?
No, I just asked like if the sequences on the alleles were different.
If the alleles, yes, basically.
Okay. So
yes.
they're just different on every person.
For particular traits.
Okay.
So for, let's take you and I for example. We both code for melanin in our skin pigmentation. I still have the code.
Okay.
You just express it more than I. I don't express it hardly at all.
Okay.
Okay?
Okay.
Like, this thing paints a lot. This is why it's like combinations still in all sense.
Yes. And so that's actually a bit more complex when we get variations or blendings. Okay, so we're not all just black, we're not all just albino. Okay, there's a blend or a combination. So that's actually a little more advanced, but you know that's a tangible thing that we can see. And mixing colors, blending is what I just tried to say. All right, so before I move on, are you okay with what a gene is? What is a gene? What does it do, or where is it found? It's a sequence of DNA that codes for a protein, and it is found on individual chromosomes. You guys further identified that alleles are basically variations of genes. Okay, so some traits do have alleles, and that's the kind of magic behind reproduction, sexual reproduction, where we're mixing and blending alleles to get something, an offspring that's uniquely combined. Where you guys were going is that all allele expression isn't as simple as black or white, on, off, something like that. Okay? There may be a variation in there, a gradient, basically. Does that help a little bit? All right, so in this image, the little red lines are just highlighting where a couple of alleles are located between on chromosome number 15 that deal with eye color. And so it's actually looking at, there's a gene, an allele variation that codes for brown pigmentation. And there's an allele that codes for blue pigmentation of eyes as well. And they're located on chromosome number what? Can you see? Chromosome number 15. And the human dictates eye color of the offspring.
Where exactly are alleles coming from? Like alleles from the parent to the mother and then alleles from the father?
Yes. Yes. Were you going to add something? Mm-hmm.
Will you alleles cross over?
They may. Yeah. Before I address that, more commonly, you'll see crossing over at the ends of the allele. And so if you look where eye color pigmentation is at the end of the chromosome, so you can see some crossing over and mixing up of this offspring, the babies, right? Where does eye color come from? You know, just kind of sneaking in traits, if you will, to kind of give some genetic diversity. Something to add? All right. So a gene, I just want to make sure we're okay. A gene is just a name for a particular trait, right, or protein. What we're learning is there may be variations of that protein. We'll call those alleles. So that I feel like we're kind of on a roll, so I just want to add you, I want you to add a little bit of detail about the alleles, which was asked just a moment ago. Some alleles are dominant, and some alleles are what? Recessive. Just a little carrot out there. I don't know if you can find that. So alleles are just variations of genes. Some of those alleles are either dominant or recessive. We're just putting those words out there. We'll fully address it better. So, I illustrated. So allele combination. That's why you'll see, for example, hair color and eye color. You notice the alleles for eye color often carry together with the alleles for hair color. And that's what these are actually kind of grouping right together. They're often carried together. So a lot of times in the human race, we'll see blonde hair, blue eyes, or dark hair, and then some other color eyes. They call it non-blue. Okay, so.
So that's like when I had like light hair. And I realized, you know, I have really dark hair. It's
That's a common developmental trait. I'm not sure what.
In the sun or anything like that? I mean, does the sun light there, or I mean?
The sun does have an effect on living organisms, and most specifically at the DNA. I'm not sure, but I don't think it has to do with that change that you see when someone's young. When you're young, you're still just growing. You're still just developing, and so some proteins are expressed and others are not. And so in that case, I'm going to guess probably you have two different alleles. One's expressed early in life, and the other's expressed later in life. But I honestly don't know what's involved with that, but it's a common thing. I don't think it's tied to the sun. I think it's just expression of proteins. Okay. Let me go back.
Let's go back to saying number 15.
Okay.
Now, each chromosome does it contain the same information?
Yes. Okay. So chromosome 15 will always code for hair color, eye color, and whatever the heck else is on there, like ATP synthase or whatever the case. Chromosome number 1 will always contain the enzymes for glucose metabolism or whatever the case. There should be no combination between, say, chromosome 1 and chromosome 15. In that case, we have a mutation, and the offspring, the cell, baby, whatever, wouldn't be viable. Additional question, Mr. Donald?
I know that I don't know. It's like most babies have like blue eyes. Like it doesn't even matter. Like you can have blue eyes, and within months later, it develops slowly.
Yeah, so Asian, Caucasian babies do express the blue protein, but I'm not sure what's happening at the iris itself. But so blue eyes is common to Caucasian and Asian descendants. Right.
Like so, I can have blue eyes as a baby, but it just slowly goes away. And I have that.
And even it can happen as late as adolescence. So teenager years when all the hormones alter your protein expression. And so someone may have green eyes or blue eyes up till age 13, and then all of a sudden puberty hits and their eyes change color.
I have a question. My grandpa had a glaucoma.
Okay.
And so, when he put the eye drops in his eyes, and then his eyes
There's some type of chemical reaction going on. I'm not sure.
okay.
I don't know what the...
Well, I just wondered whether that would be personal, like the person, or...
Eye drops have a lot of saline solution and liquid, so something about the salt is structure dictates protein, or structure dictates what? Function. I can even restate it today. Structure dictates function. You add a lot of salt to a solution, it can change the shape of our proteins that may change the way their light reflects off. So, I don't know, that's interesting. I haven't heard of that before. Also in the glaucoma case, though, that's not normal. So everything's kind of altered in the eye. So that's probably tying right into it as well.
Okay.
Was there another question? Did I address your question? All right. So let's review. Of those 23 pairs in our body, the bulk of our chromosomes are considered to be autosomes. Okay, so how can we define an autosome? What do I have highlighted here? Everything but the what? Everything but the sex chromosomes. So the autosomes are the non-sex chromosomes. They're basically chromosomes number 1 through 22. And that's all we can find, regardless of, we're all going to have all chromosomes, but gender will not influence hair color, whatever. We're going along with those scenarios. The remaining 20, let me restate this. The 23rd chromosome is considered to be a sex chromosome. If you're looking at a haploid, there's just one. In a diploid cell, there would obviously be both. The sex chromosomes is the 23rd chromosome. state that. What was mentioned is that 23rd chromosome does dictate the gender of a developing fetus. That is that 23rd chromosome that dictates the gender of a person. What I want to clarify, though, is that's not all that it does. Write that down. That's not all that it does. There's a lot more important things besides gender determination. When we look at a diploid cell, we can see a combination of our sex chromosomes. The combination can be either way. You can get X from you always get X from your mother. You may get what from your father? Y or X. Because following meiosis, one cell splits 2 times. And we produce how many? 4. Half and half. 50% of the sperm cell will have which chromosome in them? X, and the other 50% will have Y. So a man can produce equivalently daughters or sons, or mixtures. So do the sex chromosomes have only just gender determination? Because they're going to get half their chromosomes from the mother and the other half from the father.
So, just out of curiosity, is there some men that only produce certain genders, or is that not, is it just coincidental that person would have 7 kids with all girls?
It's coincidental, probability. There is also, if you think about the production of the sperm cells are going to have 23 chromosomes in them. They'll have the 22 autosomes in either an X or a Y. Okay, let's be scrupulous about X and Y sex chromosomes. What is different? They're different. What's different about them? The Y chromosome is much smaller compared to the X chromosome. If something's bigger, it's what? Let's not even look at the genetic level. If something is bigger and I want to move it from one person to another, it's going to be what? Heavier. X chromosome is heavier. It doesn't look like much, but inside of a tiny little flagellate sperm, it takes a little bit of extra energy to move an X chromosome containing sperm than a Y chromosome containing sperm. And so therefore, it's believed, and it's been quantitated, that the Y chromosome containing cells kind of get there first than the X chromosome. And so it's all timing. So if the Y chromosome containing sperm cells get there first and the egg's not there, they'll die. And then the X chromosome will get the job done, basically. The timing, but purely probability. The timing of the woman's release of the oocyte, timing of intercourse, fertilization, etc. Additional questions? That makes me think of something. In the online section, someone posted a really good video, a YouTube video, that actually addresses fertilization, which between the Unit 3 and the Unit 4 material, we just naturally go, well, what about human fertilization? So it's called, the YouTube channel is Nucleus Medical. I think it's called Nucleus Medical. So you're going to search for them. Another really good fertilization video that accurately describes fertilization. Alright, so are we okay on what a sex chromosome is? It's just chromosome number what? And it does have a lot to do with what? Gender, but is that all that it does? No. we already identified that up till now I was illustrating haploid cells, but in our somatic cells we have not one but 2 copies of all of the 23 chromosomes. So in this case, these 2 chromosomes would be considered to be what? They'd be found at a diploid cell, and they themselves are considered to be homologous to one another. They have the same structure, but may be different. I'm gonna. What? So she was saying, what? Of different genes, or we could say alleles. The homologous chromosome, they code for the exact same gene. ATP synthase, exokinase, blah, 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 DNA polymerase, whatever. They code for the same gene, but they may have different. Did I hear it? Alleles, y'all are saying it. Our homologous chromosomes, they code for the same type of gene, same protein, but those protein sequence or those gene coding sequences may be slightly different. They have different alleles, different variations. No, if I were to quantify, I could just double up each of these chromosomes. So in this first panel, let's just say the ones that are actually numbered, let's say they come from a type. I'll hide this one. Let's say all of those non-replicated chromosomes would have come from the father. If I had gone through and duplicated all of them, the duplicates would have come from the mother. So from mom, I get chromosome 1. But I also get chromosome 1 from dad. I also get chromosome 2 from it depends on meiosis, and that's, we're going to tie back into meiosis. Because during meiosis, after replication, you may get, in a sperm cell that's going to go on and fertilize, you may get chromosome 1 from father, so where you're going in that case in meiosis, what you're saying is correct. But chromosome 2 may be from the maternal, from his maternal. And so, but collectively, we have all 1, 2, 3, 4, 5, 6, 7, 8, 9, through 23. So you're correct in meiosis, following meiosis. So homologous pairs, or homologous chromosomes, are what? They're the same chromosome, they code for the same genes, but there might be allelic variation between them. Is there room to squeeze something else in there? All of your somatic cells has all the homologous chromosomes. So in your body cells, you have diploids. So you have all the homologous chromosomes.
So all diploid chromosomes are
Yes. Maybe you want to add that in your diploid. What she said is all diploid cells have homologous pairs.
So when I read this, it looks like, because I was reading on
There's not a lot of variation, but there's enough to keep us as a human race variable. So if you and I, stereotypical looking American kids, go over to where somewhere looks completely different. So go to the Middle East somewhere, a little bit of difference. Okay, and then go to, say, the core of Africa. There's a little bit of difference there as well. And so we can see we're going to go to phenotypical visible expression differences, but there's still a limit. Okay, so we're not going to see people with, well, we do, but that's not natural. That's not genetically coded. So right, there's our range, but there's still a good combination of variation. So you're right. I mean, if I were to take my sequence, okay, and compare it, my genome, and compare it to any of you guys in this classroom, they're going to be 90-some-odd% match. Okay, there will be some slight allele expression difference between eye color and, like you're saying, height, genes, maybe genes that where you see some differences is maybe those people who are more susceptible to like breast cancer. There's a gene that's expressed in that case. So there's some allele, but really there's not as much as you might think. But we're still pretty awesome, I think. So, but I think where you're going is a lot of genes have to be pretty standard because we're collectively human. We operate the same. So the foundation of our genes, we need all these structural proteins and etc. Just to make us be human and eukaryotic. Additional questions or comments? All right, that takes us, did I address everything? I don't think I addressed all these terms in the little charts. Let me go through that real quick. We defined the term diploid. We defined the term haploid, didn't we, or did we not? Okay, so let's go back and review that. What does the term haploid mean?
It's the 23 pairs.
23 individual chromosomes that are not paired up. So haploid cells do not contain replicated, just strike that word replicated, do not have homologous chromosomes in them. Haploid cells do not contain homologous chromosomes. They have how much genetic material? Half. The right amount of genetic material. Yeah. So, in other words, there's only 20, in the human, there's only 23 chromosomes. Your haploid cells actually have.
Well, but that's not fair. Now
Yeah,
we're on.
yeah, you're taking ownership. Good. The reason I him on is because we're all just basically genetic. I'm going to use the word loosely, and it's not accurate on the meanings of our parents, right? And so they are your chromosomes. Within your haploid cells, of the 23, they were mixed up. Okay, so 1 through 23 went in one cell, 1 through 23 went in another cell. I'm going to do that 4 times. Selectively. Where I want to go, though, of those haploid cells, some of them may be contributed from your mother, or some of them may be, the chromosomes in there may be contributed from your father. Just all jumbled up. Let's do that. So we find our haploid cells in the gametes. Because those will be used for the next term. Okay, or let's define that. What is a gamete? Sperm or egg cell. In the plant, we call it pollen. It sounds pretty weird, and it sounds so intrusive when it's in your nose in the spring. Yeah. Let's just call it pollen. Yeah. Just take that home and tell your parents. haploid cells, what letter, how do we represent a haploid cell? 1N. Go ahead and write that representation down. Correct. It's represented by 2N. The letter N represents 23 in the human, and obviously that varies in different species. So the gametes can be used during this process called fertilization. What is fertilization? Sperm meets egg. And then basically, what's the purpose of fertilization? It's a reproductive process to create what? Offspring and other organisms. And mimic all of it. During fertilization, what type of cells join? We gave the specific type, haploidy-wise. What type of cells join during fertilization? 2 haploid cells join to make a single diploid cell. Are we okay on these terms? All right, I'm going to kind of deviate from the visual presentation, and I want to jump ahead a little bit and then fill in the terms on page 2 and page 3 so that you can better complete your handout number 11 over the weekend. Okay, so let's turn to page 2 of your handout, and we're going to give some definitions. We're looking at the first table about an allele trait and give a description. If you remember in our introduction earlier, we said that alleles, when there's variations of a gene, we can collectively call them dominant or recessive. How might we describe an allele that is dominant? Okay, it's expressive. What do you mean? You're right, that's good.
The brown hair allele is expressed as dominant.
Very good. Okay, so a dominant allele is an allele whose gene will be expressed whenever it's present, whenever that allele is present. The dominant allele will be expressed or shown, used. Yes, you're correct. I'm going to follow the table and let's just put it in there. Where we're going in the Punnett square, in a Punnett square, the dominant allele is represented by a capital letter. So in a Punnett square, the dominant allele is represented by a capital letter. What about an allele that's considered to be recessive?
Masked by the dominant.
If there's a dominant allele present, it is masked. So a recessive allele may not be expressed, but it's still there. It may not be expressed if there's a dominant, or if it's masked by the dominant allele. And just to restate what you guys concluded earlier is your alleles come from your different parents. Okay, so it's very possible that you can allele, or both of each. Separate scenarios, okay? Just depends on what your parents' genetics are.
So does, are certain traits just naturally recessive or dominant, or is it just the amount of
Certain
allele
traits
that-
are either considered to be dominant or recessive. It's not.
like blue eyes would be a recessive trait,
Yes.
whereas brown is dominant.
Yes.
And it's the trait itself in terms of not how much sample we have on.
Correct. So, what she is correct in stating is that the percentage of expression in the population doesn't indicate whether something is dominant or recessive. It just has to deal with when the two alleles are in combination, which one's expressed. If the two alleles are present, in which we'll call a heterozygous, if the one gene that is expressed, or allele that is expressed, is the dominant one. Okay? This is important to understand because we naturally want to presume that a recessive trait must not be expressed very often, and that's not true. As stated, the blue-eyed, blonde-haired scenario. Okay? It is a recessive combination of traits. But if you go somewhere to a Scandinavian country, is that a low proportion of people blue-eyed? Oh, a high proportion of people are blonde-haired, blue-eyed. A lot of recessive traits being passed on over there. So it doesn't have to do with proportion in their population. It just has to do with those allelic combinations. So certainly a recessive trait could be the preferred trait in express in a population. Did I just confuse you? Within a population, if parents are continually contributing the recessive traits, all the offspring are going to have the recessive traits. Until there's mating with someone who has the dominant allele, and then we get into variation. And so where my confusion is within a population, you might have a high proportion of recessive alleles expressed.
So a blonde-haired person and a brunette person can have blonde-haired babies.
They could, if the brunette person has someone in their line who was blonde, blue.
And then that recessive gene become another recessive.
Or you go to Maury and find out. Alright, so let's just review that. A dominant allele is what?
The gene that's expressed.
The gene allele that is expressed whenever it's present. A recessive allele is expressed only with another recessive allele when there is no dominant allele for that particular gene.
I'm confused though with that statement because you see families that have two, the mom and dad are both brunette and have this blonde kid. And so, can there be like a dominant allele but it's just not?
Expressed. If a dominant allele is present, it will be expressed unless there's some genetic catastrophic problem.
So for some reason, the parent's allele, like brown hair, didn't.
Well, it may be
That's like a
in the scenario that Ms. Byers asked. As we develop, our genes express differently. It also ties into the complexity of genetics. Like I said, we don't have, like someone who's brunette doesn't have black hair, and someone who's albino, right? Brown is a blend. Okay, so not all traits are just this simple, like we're going to study with Mendel. And so it gets complex. So, I mean, that's a good question, but if there's a known dominant allele, it will be expressed in the presence of a recessive allele. Let's define a couple of terms about a genotype. What's a genotype? The genetic makeup of a specific, not the entire organism, just a gene. The genotype is just the nucleotide sequence of an allele or of a gene. So it's really specific. These are like the A, T, C, G, whatever. It's the actual sequence of nucleotides for a particular gene. To find that nucleotide sequence, we need biotech equipment, okay, be able to see the sequence, etc. So we may not be able to actually determine, we can now, but you, right, when you extracted your strawberry DNA the other day, oh, banana, okay, you changed it up. You could feel the DNA, but you didn't know the sequence of the nucleotide. Okay? So the genotype is just the genetic sequence of a particular allele. However, what you can tell is the phenotype. What is the phenotype? Observable trait. Like you guys are making all the physical characteristics that we can see. Or measure, whatever, quantity. The phenotype is dependent upon the genotype. If we look at a diploid cell, our alleles obviously, if there's a variation, can combine differently. If two alleles, one from a mother, one from a father, are considered to be homozygous, what does that mean? They're matching, they're the same. They're either both what? Dominant, or they're both recessive.
Little h and big H. Heterozygous.
So those allele combinations that are considered to be homozygous mean that there's no variation in the allele between your mother and your father. So if they both had dark hair, and you have dark hair, you're homozygous for that trait. However, if the allele combinations are different, those alleles are considered to be heterozygous. If an allele combination is heterozygous, which phenotype would be expressed? The dominant. In those heterozygous organisms, the dominant trait will be expressed.
So if dad has brown hair, mom has brown hair, they have a child that has brown hair, that child will have both, will have a capital H, little h.
Possibly.
But the capital H is expressed.
Possibly. Yep. If that's the case, yes. If they're heterozygous, yes.
Okay.
There's no guarantee that that child is a heterozygous without knowing the familial background. But you're correct. If we just look at it face value, a blonde-haired woman, okay, we're going to presume that she is homozygous for blonde hair, and then we would presume that the father would be homozygous for dark hair. And yes, the baby. But we need to know familial background to say that's always the case. But you're correct. If there's a heterozygous scenario, the dominant allele is expressed.
Can we go over the table part?
Yeah. On this part?
Yeah.
I'm going to try to reset you up a little bit. So I'm going to move to the next table on page 3 to contrast dominant versus recessive traits. As noted, if I'm going to, it made me want to make a note here. This is to be utilized in a Punnett square. So this is setting us up for using the Punnett square, which is just a simple genetic tool, kind of like a multiplication table. Dominant alleles are always represented how? recessive alleles must be lowercase. On the side, make a note. For a particular gene, you use the same letter representation. For a particular gene, you use the same letter representation. So for example, the scenario was like for hair, I would use the letter H. I wouldn't use B for blonde and L for black. Because I'm talking about hair color. I want to use just H. I want to be consistent, so I don't get myself all confused. If someone is considered to be, right, so I've got the diploid. If someone's homozygous for an H trait, if they're homozygous dominant, how would I write that? Two capital letters. Depends on the trait, so let's just make one. Let's just do two capital A's. I can presume this is diploid because how many alleles did I write in there? Two. What if something is considered to be homozygous recessive, the next cell? In homozygous recessive, it's two lowercase. So if we use the same letter A, we would write two lowercase a's. I have two, so that's representing what type of cell? Diploid cell. Going to the next row, it talks about a phenotype. If something is homozygous dominant, which phenotype is expressed? Dominant trait is expressed. In the phenotype, it's just a trait, so you can just repeat the word dominant. Dominant trait is expressed. What if a diploid cell is homozygous recessive? Which trait is expressed? Recessive. Homozygous recessive trait is expressed. There's no dominant allele, there's nothing to be expressed. What if a genotype is considered to be heterozygous? How would we write that? Capital A, lowercase a. I know this is a diploid cell because I have two alleles represented. And notice on heterozygous you don't use the word dominant or recessive. It's just heterozygous. You would say heterozygous dominant and heterozygous recessive. It's assumed dominant when you say heterozygous because we know that dominant alleles take over. So the phenotype, heterozygous, we said, would be the phenotype. What do you write down for a definition of a phenotype? The dominant trait is expressed. So genotypically, there's a heterozygous trait. The dominant trait would be expressed. phenotype is not the sequence, not the allele combination. So you're not writing the letters for the phenotype, right? For phenotype, we're writing what? The dominant trait. The phenotype is not the genetic. It's just the expression. The dominant trait is expressed, is what we should write on the phenotype for a heterozygous organism. On the back of page 4 is a practice for a monohybrid cross. I want you to try to work through it on your own. If you need help with me over the weekend, just contact me through email. Okay? What you're going to do is create a legend. On your own, like on a piece of paper, just make something up, like hair color, or think about your dog or whatever. Just make one up, create your own legend, and do a monohybrid cross for the hair color of your dog, or the length of your dog's tail, or whatever. Just make something up, but just be consistent in how you're setting up the tail. I want you to try to work through the Punnett square, and I'm also going to give you a dihybrid cross, to kind of work through over the weekend. So we're ready to go on Tuesday. On Tuesday, we'll come back and we'll revisit about Gregor Mendel and his contribution for where we went, and we'll put it into practice with the doing the Punnett square. I'm going to stop here. I'm going to give back the test as well as your handout number